Hi, my name is Edna, and today I'm going to tell you about our research in which we were trying to figure out which commercially available drone sensor worked best for monitoring harmful algal blooms. And if you want to learn more about this research, it is available in Limnology and Oceanography Methods and was published in 2022. So just a very quick introduction to harmful algal blooms. So phytoplankton are the base of aquatic food webs, and under normal conditions, we would have several taxa present, including green algae and diatoms and cyanobacteria. But under eutrophic conditions in which a lot of nutrients are making into the water due to agricultural fertilizers and industrial runoff, there can be an uncontrolled growth of cyanobacteria, which can lead to a bloom. And cyanobacterial blooms are potentially really detrimental to aquatic ecosystems because they can produce taste and odor compounds, which can decrease the palatability and marketability of aquacultured fish as well as water, because they make these products taste and smell kind of muddy and very unpleasant. And cyanobacteria can also produce cyanotoxins, and these can be really detrimental to aquatic organisms, as well as pets, livestock, and humans. So we want to make sure that we know when there's cyanobacteria in the water so that we can prevent these negative effects. So sometimes blooms can be really easy to identify. Here's an extreme example in a reservoir in South Africa. Here's an aquaculture pond in Alabama. And then here's a not so and also extreme but not so uncommon scene, which is aquaculture ponds for commercial production in West Alabama. And we work really closely with our farmers. And so we actually go out here once a month and we collect point samples and we put these samples under the microscope and figure out how much cyanobacteria is present relative to phytoplankton. Or we run pigment analysis, which gives us an estimate of cyanobacterial abundance. But going out there and doing these estimates on a daily basis is really challenging for our farmers because they don't really have the training and the resources available to do this. So what I usually tell them when I go out there is you can do the eyeball method for figuring out how much phytoplankton is in the water. So on the left-hand side, we have an example of a phytoplankton dominated pond. We tell them that usually these ponds are going to look camo green, and that's because they have an abundance of chlorophyll. And chlorophyll absorbs light in the green, in the blue and red region of the electromagnetic wave spectrum, and they reflect light in the green region of the electromagnetic spectrum, which is why plants are green. Now, in a pond that's dominated by cyanobacteria, we usually tell our farmers to look for a John Deere green color and to look for these areas with a lot of scum, which is usually caused by microcystis, which is a toxin producing cyanobacteria. And the reason why the pond looks John Deere green is because cyanobacteria possess physocyanin, which is an accessory pigment that absorbs light in the orange region of the electromagnetic spectrum and reflects light in the blue as well as red region. Okay, and this reflectance of blue light is what causes cyanobacteria to appear blue-green, giving them their common name. So we wanted to figure out, okay, well, we know that there's these basic spectral differences. Can the drone detect this? So this is going off of how, for example, satellites are used for monitoring aquatic ecosystems. So satellites are commonly used for monitoring the ocean and figuring out how much phytoplankton is present there. And the way it works is the sun is going to hit the surface of the water, and the algae are going to, or the phytoplankton are going to absorb blue and red light, and they're going to reflect green and a lot of near-infrared light. And then what you can do is you can take advantage of this difference in reflectance and absorption and use it to calculate a vegetation index or a band ratio algorithm. And the most common one is the Normalized Difference Vegetation Index, or NDVI, which takes advantage of this difference in reflectance and absorption to tell us or to estimate the amount of photosynthetic activity in an area. So here, all the areas that are shown as red have the highest photosynthetic activity, and these are agricultural fields. Now, a lot of these methods have been designed for either terrestrial systems, for agriculture, or for ocean marine systems, and mainly using satellites. So we really wanted to figure out, okay, well, how do we use drones for this? And that was our first question, and we had no idea which drone to buy, which one worked best. Should we spend a lot of money? Should we not spend a lot of money? So This study is literally us just figuring out which drone sensor we should use. So we had 4 sensors available to us. 2 of them were the visible light spectrum RGB sensors on board our Phantom 4, which is this one, and our Phantom 4 Pro, which is this one here. And here we have the absorption and then the different wavelengths. And here, this is the spectra for chlorophyll, and this is the spectra for phycocyanin. So you can see how they overlap with our measured wavelengths. We also use a modified multispectral sensor. No, this is a MAPIR Survey3W near infrared green blue sensor. It's basically just a GoPro. So it originally measured blue and green and red, but then they put a filter on it, so it sacrifices the red wavelength to then measure near infrared. And this is a pretty inexpensive option. It's about $400. And then this is our multiband multispectral sensor called the MicaSense Parrot Sequoia. And this is a really nice sensor because each one of the green, red edge, and near infrared wavelengths has its own sensor. So it has its own dedicated sensor for each wavelength, which is really nice. And this is a little bit more expensive, so about $4,000. We then took our images collected by the drones and we calculated 26 vegetation indices. Now, this sounds pretty aggressive, and the reason we did this is because we honestly had no idea which one of these vegetation indices would work the best. And we used a lot of indices that were originally designed for agriculture because, again, we were just trying to figure out methods. And to be as robust as possible, we collected data from 54 ponds. So some of the ponds we collected on multiple times of the year so we could get them at different algae concentrations. So you can see this is winter all the way to summer. And then we also wanted to make sure that we collected data from ponds that were really green, ponds that had visible scum, ponds that were very turbid and brown, and different sizes. So we have, I think, 5 additional ponds than the ones that are featured in this picture. And in total, we collected 72 water samples. We made sure that we collected samples from when the scum was present, as well as areas without scum. Some of the samples were collected in the middle of the ponds, but all samples were collected up to where we could see the Secchi disk, because theoretically, you can see as deep into the water column as you can see a Secchi disk, for a total of 72 samples. So here we have our results. So I'm going to show you the best vegetation index, which is here on the y-axis, for each of our 2 pigments. So I'm going to start off talking about chlorophyll on the x-axis, and then I'm going to show you the individual sensors. So for the Phantom 4 and the Phantom 4 Pro, the best index was the color index of vegetation extent, so CIF. Here's the formula. And we were actually really excited when we saw these data, because even though this isn't a perfect correlation, this is pretty darn close. And we weren't really expecting to get very good data from this very inexpensive sensor. On the contrary, we got very poor data from the MAPIR survey. So this is the best vegetation index for this particular sensor, which is the Enhanced Normal Difference Vegetation Index, ENDVI. And the correlation is very poor. The data were very challenging to process and very time consuming. So we definitely would not recommend using this sensor for this particular application. And then on the contrary, the multiband multispectral sensor did really well. And we were able to calculate NDVI, which generated the best estimates. And with an R-square of 0.78, we were really, really excited, because we weren't expecting the drone to perform this well, especially in those ponds that had really, really turbid, really brown water. It still fit the regression very nicely, and we were very excited. Now, for the phycocyanin, the drones had a harder time estimating phycocyanin in the system. So for our Phantom 4 and Phantom 4 Pro, the best vegetation index was this green-red ratio index. It's not bad, but we weren't as excited. Again, we had a lot of issues with this particular sensor. Again, the ENDVI, which is the best index for chlorophyll, was the best for phycocyanin, which is not great. And then we had, again, pretty good relationships between the data from the multiband multispectral sensor and our measured phycocyanin. And I want to point out that we don't really trust our phycocyanin data as much, because as you can see, none of our sensors measured this reflectance this absorption peak at 620. So we think that what's happening here is that it's actually, it's just phycocyanin is correlated to chlorophyll. So it's estimating chlorophyll at the end of the day. So we would say some of the main advantages of using the drone is, the main one is that you can account for the uneven distribution of phytoplankton cells in a system. So particularly when you're working with cyanobacteria, wind and wave action will push all the cells to one corner. And if you're always sampling the same area and it happens to be upwind, you might be severely underestimating how much cyanobacteria you have in the water. Another advantage is that it's really easy to learn how to use and process drone data, and you can cover a very large spatial range depending on which UAV you have. Some of the main limitations are weather. So you're limited to flying in the morning and afternoon because you don't want to get the sun reflectance on your images like I did in this map right here. And you're also limited by wind and temperature. So for wind, particularly in this flight plan, the wind pushed the drone off of its desired flight plan, and then it made it really hard to stitch these images together. And then you can only fly the drone when the temperature is above freezing. And then the other really important limitation is that the drone cannot estimate the concentration of toxins or off flavors, and it cannot tell between toxin producing species. But for the most part, we found that visible and multiband sensors are reliable for estimating chlorophyll, not as reliable for estimating phycocyanin. And we do need a sensor that can actually capture the unique spectral features of cyanobacterial pigments. And with that, we would like to thank you. And I would like to thank Catherine Cruz, who was the RU student who helped me start this project up. And I hope to see you at DASM. Bye.